So we'll go live now.
Okay.
Giri sir?
Yes.
Yeah,
Can you hear
okay,
me?
I'm starting
Okay.
now.
Okay. So shall we start? Okay. Shall we go ahead, Joe?
Yes, sir, we are live now.
Oh, okay. Good evening, everyone, all the viewers in YouTube as well as in Facebook. Welcoming you all to the 22nd session of the Vertical 3 practical sessions, which is concentrating on open and proprietary softwares. As I already informed, the sessions are from 6 p.m. till 8 p.m., which includes Q&A session. Myself, Giri, one of the coordinator for Drug Discovery Hackathon 2020. As a disclaimer, please do not share any personal details via the YouTube chat or Facebook chat during this online meeting. The registrations have started, and you should have a login account with MyGov in order to register for Drug Discovery Hackathon. By next week, you will have more updates on the CDAC Drug Discovery Hackathon virtual tool room, including registrations and others. So you will be get to know more about those details, how to get access to different tools from the partners. All of you kindly type your questions in the live chat, and we will try to address most of the questions during Q&A sessions. Certificate details will be shared only at the end of the training program, so please kindly be patient on that. Today, we are looking at two different sessions from Schrodinger, and we have the speakers Prajwal as well as Vinod. So they'll be focusing today on homology modeling as well as on pharmacophore database screening. Please keep in mind the first session on homology modeling will be a separate YouTube live, which you're watching right now, and the second video, which will be on pharmacophore database screening, on the same YouTube channel, MHRD Innovation Cell, but it will be a different video link. So kindly, by 7 o'clock or once it is finished, we will switch over to the next video. So please keep that in mind. So this session or this particular video live will get ended once a homology modeling session is ended, and we will start a new link within the same channel for pharmacophore and database screening. So please keep that in mind. So please do not go anywhere. The session is still 8 o'clock with two different topics. So over to you, Prajwal, as well as to Vinod. Thank you.
Yep. Thank you, Dr. Giri. So am I audible?
Yes, you are.
Okay, great. So
You
I
have to share
just,
your screen.
yep, I just share my screen, and that will be fine. Like I just want to share everything.
Yeah.
Okay, so what about now?
Yes, we can see your screen.
Okay, great. Thank you very much. So first of all, thank you very much, Dr. Giri, for giving me the opportunity, as well as Drug Discovery Hackathon team. So for this opportunity to present on the homology modeling part of drug designing. So today I'll be mostly focusing on template based drug designing. Template based homology modeling of a protein structure. You probably already had a few sessions on homology modeling, particularly by using different software. So I assume you might be much more aware of the basics of homology modeling, as well as why it is necessary to do homology modeling and how to perform, what are the steps involved in it, and what are the things which you could consider, or I will say like you need to consider. There are a few things. So I'll be mostly focusing on maybe same things but with different aspect, so that you will get the complete picture of homology modeling. And again, it is all template based homology modeling of a protein structure. And so again, another thing which I always say, like yes, it is very easy to perform homology modeling with Schrodinger. So this is what we will see with special emphasis on the method. So let's start homology modeling presentation. So first of all, you can note down my email ID in case of any other queries if you have in future, if your questions are not addressed in this session. So you can always write me. Please feel free to write me, and here is my mobile number also. You can always note it down. So proceeding further. So again, this is my as usual slide, which talks about basics of drug designing, or how do drug or any inhibitor binds to the receptor. And you know, it follows lock and key model, and our whole idea is to find out the key, best key, and best key means your best drug molecule. So which is so this complete methodology is heavily relied on the structure of ligand molecule, which is your drug molecule, and other thing is other thing is protein structure. So means because both information is needed for the computer aided drug designing. So imagine you have information about ligand molecule, because there are many such databases available for ligand molecules. But let's say for protein, so for protein also we have many such databases in which people used to they put experimentally salt structures like crystal structure or NMR structures. But what if if the information about structure is not available with you? Structure means a receptor structure, specifically the protein structure. So how to proceed with it? So as I have already explained in my previous lectures, like while designing a project, you need to think of a few factors, like what is the information available in literature, from experiments, from researchers. So that based on that information about your specific target, about your specific pharmacological area on which you want to design your project. So based on that, you need to first look into literature or any other source like databases or wherever is possible. And based on that, you need to decide what are the tasks which I need to do in my drug designing project. So here I am specifically focusing on the condition when you don't have the structure of protein available with you. And imagine like if you don't have structure of it, and you don't have the information about ligand also related to your protein, so you will end up with the fourth quadrant in which you have no information available. So the idea is from no information available means you need to go to a few sequence databases like Uniprot, and from there you can find your amino acid residue sequence of your protein of your interest. And by using homology modeling technique, we need to build homology model of protein. Once you get the homology model of protein, you will shift to your quadrant in which you have information of protein. So then you will have sky open, and you can work on your project. So this is the way one can proceed further. So once you will have your structure with you through homology modeling, we can basically perform many tasks on it. For example, once you will have protein structure, you can refine your protein structure. You can identify the possible binding pocket of your protein, so that on that particular pocket you can dock many such ligands and try to find out which is the best possible one. Using this tool, you can also refine the crystal structure or experimental structure what is available in the database. Like you can also refine that structure for further molecular modeling methodologies in your CAD project, or you can perform MD simulations on it to refine it further or to study it further. So coming back to the protein structure again, so this is very preliminary information about protein structure. Like protein is made up of amino acid residues, and you will have FASTA sequence for this protein. So basically this we call it as a primary sequence whenever it is denoted in alphabetical way. then it comes to the secondary structure, in which it gets into specific structure like alpha helix, beta sheets, and also there are distorted structures which we call it as loop. And then all these secondary structures come together and form a specific tertiary structure which has specific activity, means it has a biological role, some significance. And sometimes even those tertiary structures come together and form the tertiary quaternary complex, which you can see like it is basically many proteins come together and form a bigger complex, that we call it as a quaternary structure. So here the idea is we can basically build either tertiary structure as well as quaternary structure based on your requirement. So for structural determination, as you might be already aware about, there are various methods. For example, X-ray, crystallography, NMR method, and cryo-EM method for structure resolution, for solving basically the structure of your target, that can be protein or DNA or RNA. So it has specific requirements that we can discuss it in more detail if you are interested in. But in short, like there are various methods of structural determination that you can find it out. And there are many such databases, for example, Protein Data Bank and Protein Data Bank of Japan and Europe. So I mean, there are several databases available where experimentalists used to deposit their structure, and these are publicly available databases where you can simply go and search for your protein of your interest and see if it is already solved and available. I mean, the coordinates are available for you to use it further. So don't forget to browse through those databases before proceeding for homology modeling studies of your target protein. So the question comes like, what if we don't have a structure of your protein, if you don't find it anywhere else? So what you can do? So whether is it possible to build a structure of protein? So usual answer will be yes. But for that, the minimum information which is required for you is the amino acid residue sequence of that particular protein. And from where you can get it? You can get that information from basically a Uniprot. So that's the sequence database. It consists of a lot of information about the protein. So if you see about homology modeling, so basically name itself indicates something. It starts from homology. So homology means nothing but it tries to identify which is the homologous protein or homologous sequence. So if you find that homologous protein, so based on that homologous protein, you can build the protein structure of protein of your interest. So you just need to remember one fact, that if there is a sequence similarity, then there can be a structural similarity. So as you want to look for the structure which you want to build, you need to look for the structure which is you need to look for the sequence which is similar to that of your target sequence. So if you find that similar sequence structure, then of course you can build the structure of your target protein. So for that, it is important to have a template structure. So template structure is nothing but the structure of homologous protein, which can be available in experimental protein data banks like like PDB or Japanese PDB or European PDB. So there are a few steps which are involved in homology modeling, so that you need to know, like what are the steps. As I already explained little bit about homology modeling, means it is to find out the homologous protein. So and first minimum requirement which is there is your target sequence. So here onwards, like whenever I say target means the protein for which you want to build a homology model, and template means the crystal structure or any other experimentally reported structure of protein which you are going to use it for building the homology model of your target sequence. So these are two terms, target and template. So in general, the homology modeling protocol includes, first of all, the retrieval of your target sequence of your protein basically. So that you can get it from Uniprot database, and we will see how. So once you will have sequence of it, you need to perform some blast searching on it to identify the suitable template that can be mostly based on the sequence similarity or sequence identity, or there are various factors which you need to consider before selecting a template structure, suitable template structure, like sequence identity, similarity, then query coverage should be maximum, then there should not be many missing residues in the template structure, then what is the resolution of that structure, then whether it is present with a ligand or without ligand. So with ligand is always preferred in drug designing. And other thing can be how much is the activity of that inhibitor which is bound in the protein template structure. And so these are the factors you need to consider for the template. And then once you choose your template, you need to align those target and template sequence. So for that, you can choose like single sequence alignment, means using single template, or you can use multiple template, or you can also use secondary structure prediction method to predict the secondary structure of your target sequence. So once you finalize your template structure, you align it, and then the last step is model building. So we build model using prime is the module to build homology model. So there are various methods, like for example knowledge based method, which works when you have high sequence similarity. Then there can be a energy based method, means based on energy of a homology model, it choose which model is the best out of it. Then you can also use whether you want to incorporate that ligand which is present in template, or ions, whether ions has biological significance, that also you can consider. You can also consider if there are water molecules, important water molecules present in the template structure, that also you can build it. So other than that, you can also predict various side chains, and you can fill the loop if it is present in if it is absent in template structure. So you can also build that loop newly. So you can also mutate few residues if you want. So whatever model you get based on all that methods, you need to basically check that model and validate whether it is right or wrong, or how much correct it is, or whether it needs further refinement. So once you build that model, you can check it using for example traditional methods like Ramachandran plot or Irad plot, or checking of various steric clashes or RMSD between target and template. Or so to consider all those things, we have a very I would say very compressive tool which is called as protein reliability report, which gives you it looks like this particular image, and in just one view it gives you all information about where are the problems in your protein structure that we are going to see it further. And based on that, you need to decide whether it needs further refinement or not. So if it is okay, then you can proceed further with that homology model that it is correct. If it is not good enough, you need to go for refinement. So that can be performed using various methods like sidechain prediction, loop refinement, energy minimizations, and we try to generate a more reliable model. So basically this is the iterative process, like building model, validation of your model, and model refinement. So that all goes in iterative manner, and based after many iterations probably you will get best structure out of it, and that structure you can further use for the computer aided drug designing methods, so for further molecular modeling studies. So this is what the in general protocol which you need to follow. But there are few things which you always need to keep in mind, like I would like to focus specifically on very few things, not many, but there are many such things which you always need to consider while building a homology model. So the first which is very important thing which you always need to remember or take care of is your template structure. That template structure should be good enough with some good resolution, no missing residues, good query coverage, and good identity and sequence similarity. So that template structure should be good enough. So if you get a good template structure, so most of your problem get already solved. So, but if you don't have good template structure, then many things come into picture that you need to take care of. Then once you get even a good template structure, your sequence alignment should be best enough to build a better model. So for example, you can see here in the image, like you have target sequence and template sequence. So basically sequence alignment means the both sequence, it will try to align, and wherever it aligns, it simply copy that particular secondary structure from template structure to the target sequence, and in this way it builds the homology model. So that's the reason why the sequence alignment should be very well enough for optimum. Other than that, one more thing, like force field. So force field, most of the people ignore that thing, but force field is very critical parameter in homology model building. And I would say not in homology model building, but in all molecular modeling study, force field plays very important role. So it should be very optimum, good enough to build your model to give you best or possible or reliable results out of it. Then there are other things like like globular proteins, they stay in the solvent in biological environment, so you should consider that solvent environment in it. You can consider explicit solvent or implicit solvent, but it should be more reliable. And there might be a case when you are working on membrane bound protein, for example GPCR proteins or ion channels. So whenever you try to build that such protein structure, so you always need to consider like it has a membrane environment, because as such that in biologically those proteins are embedded in the membrane. So one should consider that membrane implicitly or explicitly while building a model. So all those things are taken care very well manner in Schrodinger. Other than that, the loop sampling is also one of the critical parameter, because many crystal structure has missing loop regions, and in such cases we need to build it from ab initio, and you never know how long the that missing loop will be. So you probably need to build a loop, let's say starting from 5 amino acid residues to 20 amino acid residues, and it should be reliable enough. So these are the few critical things which you always need to consider while building a homology model. That's the must do thing. So talking about good template structure, it's all depending on the condition, whether you want to go with single template modeling, or you want to use multiple template, or from multiple you can build a consensus model out of it. Means basically you will use many different templates to build a single homology model, and that can be used based on consensus modeling. So there can be other thing which is chimeric model. So for example, there are two structures which are very closely related isoforms, and there is a difference of only 10-15 residues between target and template. So in such case, basically it is considered as a chimera. So you can build those chimeric models also using Schrodinger suit. So not only that, but if you are somehow dealing with the quaternary structures, which I have told you before, like many tertiary structure protein structures come together and they form a quaternary complex. So for example, dimer or multimer or trimer, so those things you can also build using homology modeling. If you are specifically working on GPCR proteins or membrane bound proteins, so those protein needs very special treatment, because those are membrane bound protein, and as such if you see, it has very low sequence similarity that can be less than 30%. So in such case, like, because the structural fold is conserved, so it needs a different treatment. It is not at all same as that of globular protein. So software should consist of those software should treat the GPCR models as a differently. So we have such a option in which you can consider this membrane bound protein specifically in different way. Other than that, we have advanced loop modeling. Means you can build a model for, let's say, starting from 5 amino acid residue to 20-30 amino acid residue long loops. You can build it from ab initio. So these are the things which one need to consider whenever you are thinking of template based modeling. So there are various options available with us, and we can use those options to build a reliable homology model. Then next thing comes to picture is force field. As I always say, like, force field is very critical in molecular modeling studies. So your force field should able to predict the exact interactions, say non-covalent interaction between amino acid residues or packing of the structure in very specific way, like what is in correct way, I would say. So for that, the force field is very important in such cases. Like, it should consider all that confirmations of various amino acid residues and their packing in very correct way. So that's why, I mean, until and unless you have correct packing, you will not get the correct structure as output. Then another thing comes into picture is loop sampling or loop modeling. So with Schrodinger, you can have a very crucial protocol for loop modeling or refining loop structure. So here you can see there are various methods basically which you can use it for the loop sampling. So those methods can be cooperative loop sampling or serial loop sampling. So what does it mean by cooperative loop sampling? Means cooperative means all together. So basically, for example, in your structure homology model, you want to refine loop, let's say 5 different loops, which might lie near to each other and they might affect on each other. So in such cases, like you can go for cooperative loop sampling in the way like they all get refined at the same time. Means it also consider effect of each other. Effect of each loop on all of those loops which you are refining. So means all loops are getting refined together. So that is called as cooperative loop sampling. And let's say if you are refining again 5 loops, but those are not near to each other or they don't have effect on each other. So in such case, you can go with serial loop sampling, in which basically each loop will get refined one by one, means one at a time. Basically serially. So these are 2 different type of loop sampling. So I would say so far my knowledge or my experience considering this method is really helpful if you want to build a if you want to sample a correct structure for the loop regions. Then there is another method which comes into picture is basically default extended or ultra extended. Means it depends on the number of residue which you want to refine. So for example, if you are going to refine less than 5 amino acid residues in the loop, then you can use that default method which is followed by most of the software. But if you need to refine more residues like 5 to 10 residues, then it needs really better treatment or better sampling of that loop region, and that is covered in the extended loop sampling. And even if it is more than 10 amino acid residue, that is getting covered into ultra extended protocol. So you can basically use with these various methods for the loop sampling, so that all options are present here. Other than that, as you can see here, there are various type of implicit environments. For example, default means nothing but the solvent environment. So most of the globular proteins which are present in solvent environment, that are getting covered in default implicit solvent method. But if you are dealing with membrane bound proteins, so you can also consider that implicit membrane environment for your protein structure or building or crystallographic unit. So if it is present in crystallographic unit. So these are the various things which are pretty well covered in the Schrodinger suite. So other than that, as I talk about the explicit solvent, so so in MD simulations, for example, you use explicit water molecules to treat how proteins behave inside the solvent. But because it is computationally expensive method, so there is a gateway out of it, which is called as implicit solvent consideration. So we consider that implicit method, and we have implicit model. So that also performs very well. And as you can see in refine loop, you can basically refine various loop regions at a time using cooperative loop sampling or serial loop sampling. Considering this loop refinement method using this implicit solvent method, you can see there is a statistics of let's say this loop length, so which starts from 14 amino acid residues longer loop up to 20. So you can see it's really longer loop. I mean, unusually it needs really different treatment for considering that loop, basically loop sampling. So you can see here there are many cases for each number of loops. So let's say for 14 amino acid residue longer loop, there are 36 cases, and for 15 residue longer loop, there are 30 cases. So for, they tried to build this loop structure for these many longer loops, and they found the RMSD of basically backbone RMSD, which lies within even 2 angstrom of the actual experimental structure. And you can see like there are number of cases, like 91% of cases which fall within 2 angstrom of RMSD. And so this statistic itself says how reliable this method is for the even longer loops. So normally many softwares miss this information, but I would say this is very, very important information which you need to consider while building a homology model. So other than that, we have a tool for side chain predictions or using various type of environments. Like as I already explained, even implicit membrane can be taken care of. So while predicting sidechain, it is again, as I said, like force field is very much important in dealing with all those things, as well as solvation models is also important thing which we use. VSGB, this is a very, very critical solvation model, implicit solvation model. So here there is an example of there are interaction between two different amino acid residues, and it is optimum inside the protein structure. So one need to basically generate that interaction if you are building a good type of model. So there should be internal hydrogen bonding network that should be optimum enough. So that can be only possible by use of very refined force field, very good implicit solvation model. So those both things are critically covered in this particular panel, like solvation model and force field OPLS3E, and we can predict various conformation of side chains in very nice way. Other than that, means as I said, like there is a protein reliability report and other information about protein structure, which says Ramachandran plot. That's very classical information through which you can evaluate your model whether it falls within acceptable range or not, or there are various type of deviations or steric clashes that can be also reported in the protein report panel. And in just one go, you can see like what are the deviations present in your structure. It can be your steric clash, it can be your bond length deviation, maybe bond angle deviations, and many other factors. In specifically protein reliability report, in just one view, you can see like wherever you have problem in your protein structure. Like for example, wherever it is green means it is very well enough, but wherever it is red in color means you need to take care of it. So for example, in this particular example, it has red color on steric clashes, means there are many steric clashes present in this structure that one need to take care of it. And yellow means you can consider that one means it seeks attention, but red, that you need to resolve. So these are the things which you can basically you need to take care of while building homology models. So there are various challenges which I found like it is worth to discuss about it. As I always says, like the alignment target and you should have suitable template structure and they're very well aligned. So if you get high sequence identity or similarity, then most of the problems will get addressed. So you really don't need to worry about the quality of homology model in such case, because it will certainly generate high quality of structure and in very fast way. But if it is low sequence identity, then yes, you need to have manual intervention and you need to manually do a lot of things to get it done. But fortunately, all those things are pretty easy and very nicely covered in the Schrodinger suite. Then other than that, for refinement of model, one need to refine loop structure, one need to refine side chain conformations, or specifically binding site conformation that you always need to look for, and the packing of all those amino acid residues or optimum internal hydrogen bonding network. So all those things you need to always take care of while generating homology model and while evaluating homology model. So these are the things. So so I would say that like limiting factor where it is, so it lies within the sequence similarity. So let's say if you have high sequence similarity like more than 75%, then everything is fast enough and better quality of model. But if it is in 50 to 75, then you will get into good type of model and you probably need less human intervention, but still your model will be good enough. But if it is getting low and low, like 30 to 50, then certainly you need to manually did the alignment and you need to perform various loop modeling to check whether you will get the better model or not. But if it is less than that, then yes, then all things come into picture that at each and every step you need to critically analyze your model. You need to check each and every I would say the sections of your particular model, that which loop is deviating, how it is behaving, which part of your structure is good enough, which part is not good enough, and how to solve it. So these are the things which you need to think of while generating homology model. So in the simplest way, with Schrodinger suit, we will see like we'll try to generate a homology model of human phosphoglycerate kinase 2, and which will be the based on the template structure of human phosphoglycerate kinase 1 structure, which is available in protein data bank. So now in next exercise, we will see how to perform homology modeling of human phosphoglycerate kinase 2 protein structure. So before proceeding for homology modeling, I must need to understand whether any crystal structure or any experimental structure is reported for this particular protein or not. So the best possible way to see it is I would say like Uniprot sequence. So Uniprot database. So you can always go to uniprot.org. So this is the database which consists of all information about sequence, and then I will just type the protein of my interest, for example, human phosphoglycerate kinase 2, phosphoglycerate kinase 2, and that can be my query. And once I try to search this particular protein, I will get a lot of entries as a result of this query search. And in this search, like because it is text based search, so it will give me a lot of results, and I need to find out where is the protein of my interest. And as I said, like I'm looking for phosphoglycerate kinase 2, and that will be from human. So this is the entry which is of my interest, which consists of around 417 amino acid residue. So I'll just open this particular entry, which has a ID of P07205. I'll just open it and see what is mentioned here. So there is a lot of information available about this particular sequence. Means everything, I would say, where it has role. On left hand side, you can see every information is present, like function, what is taxonomy, where is the location, what is pathology or biotechnology of this protein, where it is involved. But specifically my interest is whether the structure is available for this protein or not. So I'll just go to the structure section and see what is written here. So it gives me the link of various 3D structural database, and currently there is no link present here. It means there is no crystal structure reported for this particular protein. So next thing comes into picture, like, yes, I have an opportunity to build a homology model of this particular protein. So I need sequence. So for that, I'll just go to next section, which is called as sequence. And in sequence, I'll find that particular sequence, which consists of around 417 amino acid residue, and what is the mass of protein and when it is deposited, and all other things are information is present here. And what I am interested is in sequence and this FASTA file. So I just need to download this particular FASTA file and store it in my local directory somewhere. And I just need to save that file. So I have already saved this file in the directory, and now so everything is clear, like no crystal structure is available. I have a sequence of it. So next point is we need to go to Maestro to see how homology modeling works. So I'll just start the Maestro by clicking on the icon called Maestro 12. So once it opens the panel, so it looks like that. Probably you have seen the same thing in our previous lectures, like how to see, how to operate in the Maestro visualizer. So I would directly go to the task, which is called as homology modeling. So basically there is a task button on bottom right. So you just need to go to task, browse. And as you see, like many times I mentioned about prime module. So prime is one of the module which is responsible for protein building. So you will find that prime option here, and in that prime option you will find all those options which are related to the homology modeling. For example, homology modeling or side chain prediction or loop refinement or other things like energy minimization and all. But for me, the important thing is homology modeling. So I'll just click on homology modeling. It opens the panel And there are two types of method, like whether you want to go for multiple sequence viewer or you want to go for structure prediction wizard. So along with these names, it also gives me the hint like which option I should go with. So for example, this particular secondary structure prediction wizard, I got a hint like this is ideal for those who may be new to homology modeling and unfamiliar with the process. So if you are a new user, then you can go with this option. But if you are professional enough, you can go with the first option. So I'll go with the simplest method, which is called as structure prediction wizard. So I'll just open this panel now. And so this panel is pretty simple. There is nothing here. It just says like get sequence from. So as I said, like I have already downloaded the file FASTA sequence and store it in a directory. So I'll just go with the file, or if you want to copy paste it, you can also go with text, or you can also mention PDB ID or something else. So I'll just go with the file option, and the file is present here with the name of human phosphoglycerate kinase 2 protein. So that's the FASTA file. I'll just open that file and it gives me all information which is present in that file. So you can see like what is the ID of this sequence, what is name of this sequence, what are the amino acid residues present in it, and you can realize like wherever I move cursor, it is interactive enough to tell you what is the residue number, that you don't need to count it separately. It directly shows you what is the residue number and which the position it is. And the same information is also available here. Yes, this is the sequence which I am interested in for building homology model. So I just need to click on, once this panel is done, like you have imported your sequence, that's it. If you want to edit this particular sequence to produce mutants to chimeras, you can always use this edit option. But because at this exercise, I don't want to edit anything. I just want to prepare a homology model of this particular protein. So I just need to go to next. And in next panel, it gives me an option for blast homology search. So still the sequence is same. You will have a further option called blast homology search. So basically it connects to the internet. So in option, you can see here, there is an option to use web server for blast search. Means you don't need to store the complete protein data bank in your local directory for searching, but it can directly use the web server and try to fetch all information available in protein data bank. So you need to use protein data bank for the blast search, and that's it. You just need to click on blast homology search option. Once you click on this blast homology search option, it will fetch you all the possible available information for this particular sequence by comparing all those sequences available in the protein data bank. And here you can see the result of blast homology search. You will get it into the table manner, like all possible results are given here. You can browse through all results. It is just like a table. All information is tabulated, so which consists of ID of template structures, what is the name of it, what is chain of it, what is score, how much is the identity, how much is the similarity, how many number of gaps present in it, and whether it consists of any ligand molecule, whether it is present in it. So for example, which experiment it is solved, which ligand it is present in it. For example, in this particular structure, ADP is the cofactor which is present. What is resolution? What is the experimental method to solve it? So in this way, basically a lot of entries you will have, and so the entry which is selected here is same entry which is aligned here on the panel, and it is all colored with respect to residues. So you just need to think of like which protein template structure I should go further with. So it's easy to even browse through it, like the entry which I select, it automatically appends here in the sequence. So basically all of those structures are from the same protein, which is phosphoglycerate kinase protein. You can see here their name. So I'll just try to show you any other protein structure which has a different amino acid residue sequence, and you can see the sequence got changed. So means you don't need to align, it automatically gives you all that alignment, what is the query coverage for this particular sequence, and all available information. For this exercise, I would go with the first entry, which consists of all acceptable range of identities and similarities and no missing residues and other things. It also consists of ligand molecule inside it. So I'll just go with the first entry. So it automatically gives me the option and aligned template and target structure here. So it looks better, like most amino acid residues are identical and similar. So that's it from this panel. I'll just go to next. And in next, it shows the sequence which I have selected, that template structure whichever I have selected. And now I'll be proceeding with only this particular template structure. So because it says like it is already aligned, but you can always align it using this align button if you are not happy with this particular alignment. So because it has a high sequence identity, so whatever alignment was done by software in by default manner, it was very correct. But if you are not happy with it, you can again align it, or you can also predict the secondary structure elements of this particular structure. You can see here using this secondary structure annotation, you can find it out which regions belongs to the which type of secondary structure, whether it is helix or beta sheets or in loop region. So it is there. And as I said, like Schrodinger also has this GPCR specific treatment. So if you are dealing with GPCR proteins, which has very low sequence similarities, like less than 30%. So in that case, as I said, like it needs different treatment. So for that, we have option of GPCR specific alignment, that also one can use it for GPCR proteins. So that's it from this panel. And the next thing is the final option for homology modeling. So this is what is the final alignment of your target and template structure. So the sequence which is highlighted in pink color is the template structure, and sequence which is in a white background is basically a target sequence for which you want to do homology model. So as I said, there are various methods. One is knowledge based methods, means based on the available information about the crystal structure or template structure, it builds the model. Or there is another method called energy based method, in which based on the energy it tries to refine the homology model. So as it says, like knowledge based method, once I move mouse on knowledge based method or any option, it gives me some hint. So it also gives me the hint like when should I use this knowledge based method. So because it is very high sequence identity structure, so I could go with knowledge based method. And as I said already, there are various other type of modeling methods like chimera or composite model generation or homo or multimers or consensus modeling. So all these options you can use if you want, if you are building a quaternary structure or chimeric structure. So these are options are present. Then it comes to the inclusion of ligand or cofactor, metal ion or water, whatever is present in the template structure, that you can straightforward build it using this homology modeling protocol. So because ADP was present inside the template structure, so I have just selected that ADP so that it should be considered in the homology model building. I want this ligand to be inside the binding pocket of my homology model. So that's it. So this is what the option available, and you just need to click on build model. So once you click on build model, it runs in the background and it gives you the output of homology model for the particular structure. So for example, I have already built the homology model. So this knowledge based method is pretty straightforward, and it just give you the homology model in straightforward way, in very quicker manner. So I just close extra panels. And okay, so here you can see the built homology model. The name itself indicates that what is a sequence ID, like which is P07205, then phosphoglycerate kinase 2, and the homology model built based on 2ZGV chain A structure. So this is what the homology model. You will be surprised to see why it is shown in different colors. So the color means this dark blue color is for the structure which was built based on the template structure, means for those structure it found the identical amino acid residues in the target as well as template. So those regions are highlighted in dark blue color. In cyan color, basically those are the amino acid residues which are not identical but similar in case of target and template. And there are other regions which are mentioned in red color, means mostly these regions are loop in structure. So red color means always alert. So red color means these structures are not found in the template structure. Software has newly built this particular loop. So now it comes to the thing like which region you need to consider for the further refinement. So wherever you find red means you definitely need to take care of these regions. So next thing comes into picture, like to validate this model. So how good this model is and how to validate it. So for that there is very classical thing which is called as Ramachandran plot. So whatever model you have, so you just need to find out the Ramachandran plot of this particular model, which you will find in structure analysis section. So in just structure analysis, you will have option for Ramachandran plot. You just need to click on Ramachandran plot and it will show you what are the amino acid residues which are present in allowed region, what are present in disallowed region. And as I always say, like in Schrodinger, all panels are basically interactive in nature. So interactive means wherever you move your mouse on these points, you will see information available with this particular amino acid residue. So for example, on the top it is showing these points belong to the amino acid residue alanine. Not only that, but if you click on this particular point, it will also highlight it on the workspace, and it says like this disallowed amino acid residue is present in this red color region. So means you will have chance to refine that particular loop region, because there is one residue alanine 377 and that comes into the disallowed region. So you need to make it into allowed region. Other than that, there are various points which are mentioned in triangles as well as squares. So those are basically glycines and prolines, and those has always exceptions. So you can ignore those amino acid residues if it falls in disallowed region, because those residues have exception. So remember, those residues are glycine and prolines only. Rest of the residue you always need to take care of if it falls within disallowed region. So once you get that model done, once you identify, yes, this is the thing which you need to refine, and from Ramachandran plot, other thing which you can analyze is basically, as I said, protein reliability report. So this option is also present here, protein reliability report. You can directly open this panel. You have loaded that particular structure and just you need to click on run. So that's it. It is single click option in which you will get the all possible available information for this particular protein. So for example, I have already run this job and you can see how it looks like. So for this particular protein structure, it gives me the information in just one go. Like it has a red color patch on steric clashes, means there are many steric clashes present here. There are buried unsatisfied donors present here. There are peptide planarity which has problems or bond angle deviation. So these are the few things which you need to take care of. And how to take care of this, basically refine this structure is by either loop refinement or by using energy minimization. So if you want to perform energy minimization on that model, there is an option for energy minimization in the prime module. Like we have energy minimization option. So you can just open this panel and everything is present here, like solvation model, force field, and what residues you want to minimize. You just need to click on run and it will run in background. It will give you the output protein structure. And you can see like how this after minimization, the structure got changed. Then again, you can run your protein reliability report on the minimized model. And you can see like now the situation is changed. Whatever steric clashes were there were already removed now. But there are a lot of things which are present in peptide planarity that you need to get rid of by again either performing energy minimization or loop refinement. So for loop refinement, like you can use the same model for loop refinement. That option is also present in prime module. So you will have option for loop refinement. If you simply open that panel of loop refinement, it again gives you solvation model and force field which you can use. And you need to identify the just which amino acid residues are present in loop, which you can which you can perform loop refinement on. So there are options like you can identify simply non-template region, and you can see on the screen this non-template region belongs to this red color region. You can see it automatically identified that these are the amino acid residues starting from 374 to 385, and this range belongs to this red color which is non-template in nature. And if you remember, you had amino acid residue which has number 377 alanine that falls within a disallowed region of Ramachandran plot. So that amino acid residue also belong to this. So I mean, in this way you can basically directly click on run and it will refine your loop. Or if you want to refine multiple loops at a time, so you can go with a looking for all loops which are present in the structure. Like there are so many loops which are present in the structure, and it has ranges. So basically this software can automatically identify which are these kind of loop regions which are present in your structure, and where are the amino acid residues are of your interest which you wanted to refine. So you can just select multiple loops for the refinement by and then once you select those loops, there is an option present in it, and these options are specifically for various environments and various type of loop sampling methods, as I already explained, like cooperative loop sampling or serial loop sampling. Or you can also use how many amino acid residues you want to refine, like if it is less than 5, you can use default. If it is more than 10, 5 to 10, you can go for extended or ultra extended. So based on your requirement, all options are present. You just need to choose your option and click on run. That's it. So once you refine your loop region, so it might take some time. I got, like, after refining loop, I got 4 different structures as an output. And you can see here how the confirmation of loop got changed little bit. This red color region, basically there it generated various confirmation of this particular loop. And now you can choose which is the best sample loop out of all those structures. Again, you can use this protein reliability report or Ramachandran plot to check it. So I'll just check the reliability report of this particular structure, and you can see even after refining the loop, because it was a specific region, not all things are getting satisfied, but now there are only few, like peptide planarity, which I need to take care of, and backbone dihedrals, which I need to take care of. So with this, I would again go with like a minimization of this particular protein to see whether it is refined. So I have again performed minimization and optimization of the protein structure, and I again check the reliability, and you will see in just one go, like many things are getting better now, better than before. Like this red color peptide planarity is being reduced now. There are few improper torsions that that we need to consider again. So in this way, basically we need to go step by step, like loop refinement, energy minimization, or dihedral backbone minimization, or optimization of internal network. So there are various steps which you can perform for refinement of your model, as I said. And so in this way, you need to proceed further for homology model reliability check, as well as to refine your homology model and select best out of it. So just as an example, I wanted to check how the crystal structure looks like, which I have used for a templates, which I look for the templates as a template structure. And I just tried to superimpose both structures on the top of each other, and you'll see like where the ligand is present in target and template structure, and see whether ligand is also superimposed so that it retains the position or not. And here you can see like ligand as well as protein are basically ligand from template as well as ligand from homology model are present at a same place. Means it somehow validates the binding pocket of your homology model, that it is present at the same place with same number of interactions probably. So, and at the end, I wanted to see like how protein reliability report of this particular crystal structure looks like. So I have loaded that crystal structure and checked the protein reliability report of this crystal structure, and you can see like for 2ZGV, everything is very good in that sense, like everything is green or yellow. So it means this crystal structure is very good enough for which we have used for homology modeling as a template. So with this, finally we got a final structure out of our homology modeling protocol, and that if I am satisfied with all other requirements like all the reliability reports and everything or binding site validation, I can proceed further with this homology model. So this is all about homology modeling. So with this I would again always like to highlight our help button. Means in help and help, you will have all answers of your question. You just need to browse through it and type your question. You will get definitely you'll get the answer in a reliable manner, or otherwise you can always contact us. I have shown you so far homology modeling, but there are many such features which are present in this particular prime or bioilluminate subsuit, and that will be discussed in consecutive sessions by Dr. Pritesh. And there are many such things which are present here. Even one of the striking and most important thing which I found is you can also consider non-standard amino acid residues, and this is also very critical nowadays. You can have many non-standard amino acid residues which are present in the structure. That also you can, it will take care of. So this is what is a part of Schrodinger suite and with many functionalities. With this, I would like to acknowledge my team which are working in Bangalore, and we are working round the clock to answer your questions, to support you in as possible as manner. So with this, I would like to thank you for all of you for joining us, and specifically Drug Discovery Hackathon team for giving us an opportunity. And if time permits, like we can have a few questions, or otherwise you can always write me back questions
Hi,
on my
Prajwal.
email ID. Yeah,
Sure.
thanks.
So, Prajwal, since we are a little bit late, so I would recommend we will take the questions at the end. So, can we start with the next session?
Yes,
Okay.
definitely.
So, all viewers, I kindly request this streaming is going to end, and there is already a stream in the YouTube channel already available. So, the next session will be on that channel. This is